For farmers supplying our world with food, there's one thing they can count on: demand. We all need to eat. Outside that, nature and the elements combine to make farming a risky business. What if rain is scarce, or temperatures are too hot? What if harmful bacteria enter fertile soil? Or insects damage leaves? There's not much science can do to increase rainfall. But when it comes to crop damage caused by bacteria or insects, biotechnology is delivering new answers. Today, we take from these attackers all the genetic information needed to make plants they damage resistant to disease and insect attack. In animals, we can create new characteristics at the earliest stages of life. Milk from cows can be enhanced to provide better nutrition. The growth rate of cattle and other important livestock can be improved. And Earth's most complex organisms stand to learn and benefit from the fascinating science of biotechnology more than any other creature. We're talking about human organisms, people like you and me. In this issue, enter the living world of biotechnology. Take an inside look at the building blocks of genetics. Learn how gene splicing is accomplished, and understand why plants, animals, and people are so closely linked together. We all work to better our environment and our lives. We want to improve our world because we're more aware of its problems and have more power to find solutions than any other species on Earth. We see expanding populations and a need to grow more food. The science of biotechnology offers solutions we're just beginning to explore. With careful attention to the biological safety of genetic experiments, scientists are putting it to work for plants, animals, and people. The basic premise of biotechnology, that traits from one organism can become part of another, was discovered within our plant world. In the mid-1800s, Gregor Mendel advanced the concept that different generations of peas contain similar characteristics transmitted by elements of inheritance called genes. This early research led to a natural method of conveying characteristics from one plant to another, called crossbreeding. Crossbreeding is useful when transferring genetic traits in very similar plants. It's also useful when there's enough time to isolate and transfer desired traits over several generations of a species' lifespan. But crossbreeding has limitations. Organisms must be genetically compatible, or a transfer may never take place. It can also take months, even years, to isolate and transfer those traits farmers or botanists are interested in. These oat plants demonstrate the limitations of crossbreeding. Mildew, which we think of as harmless, becomes a damaging fungus if you're a farmer growing oats. It can ruin your otherwise healthy crop. But if you could find a similar oat somewhere in the world that was immune to mildew, and you could transfer that characteristic to your plants, you'd never worry about mildew again. With this goal in mind, botanists traveled the world, finding wild oat plants and testing them for natural mildew resistance. After months of searching, sorting, and categorizing, 2,000 samples later, they found one in Algeria. They hoped that cross-breeding seeds from Algerian oats would lead to future generations of oats strong enough to ward off mildew damage. But in this case, cross-breeding failed. Mildew-resistant genes were incompatible with those in susceptible oats. Like oats, tomatoes are also open to attack by a deadly enemy called tomato mosaic virus. Not a single species of tomato has natural immunity to mosaic virus. Immunity must be invented. In biotechnology, invention begins with deoxyribonucleic acid, or DNA. DNA is made up of nucleotides, genetic building blocks formed from sugar, nitrogenous base, and phosphate. It's shaped like a double helix. A single DNA molecule has millions of nucleotides joined together in sequences called genes. Genes contain all the chemical information that gives every organism its unique size, shape, and biological characteristics. It's a unique DNA fingerprint that makes a tomato, or an oat. DNA is a crucial element in the science of biotechnology. We then discovered plasmids, special circles of DNA that can be cut open, spliced together with genes from a different organism, and resealed. It's the scientific version of cutting paper with a scissor and gluing it back together in a different way. We also discovered special bacteria called agrobacteria. Agrobacteria carry plasmids into cells of certain plants. Think of agrobacteria as unique travelers on a genetic highway. We then learned how to grow an entire plant from a single altered cell. Scientists needed to understand all these things: DNA, plasmids, agrobacteria, and cell growth, before they could create a tomato plant immune to mosaic virus. Here's how they did it. They cut open a DNA plasmid, inserted a tomato mosaic virus gene, and glued it back together. In this drawing, you see a DNA plasmid carrying the virus gene, entering a single cell of agrobacteria. Putting small sections of tomato leaf in a liquid suspension of agrobacteria lets plasmids transfer new genes from one cell to the other. It looks pretty quiet from out here. But inside, in a single cell, biotechnology takes place. agrobacteria attach themselves to tomato cells. The plasmid, with a mosaic virus gene seen here in blue, duplicates itself. The new gene transfers over and becomes part of the tomato plant. Still, creating single pieces of genetically changed leaves is far different from a greenhouse full of tomato plants. To move further ahead, biotechnologists must be able to turn individual leaf sections into full-size tomato plants. And that's exactly what they do using a process called tissue culture. Now, when a molecule of tomato mosaic virus tries to attack a tomato plant, it finds out that it's already there, and the crop is completely immune. In other common food crops like rice and apples, the power of biotechnology reveals itself further. Scientists are changing plant size and growth patterns to our advantage. Rice plants are their own worst enemy. They can grow so tall, entire stalks are often destroyed by wind or rain. In Southeast Asia, where monsoons strike during the height of growing season, crops can be completely destroyed. But what if you could limit the height of rice? Short stalks could resist wind and rain damage better than tall ones. Using computer imaging techniques, biochemists identified rice genes associated with height and created a chemical height inhibitor. Far from damp Southeast Asian rice fields, greenhouse testing is underway. Everyone involved wants to ensure that shorter rice plants produce plenty of rice, and that the new process is biologically safe for plants, animals, people, and our environment. Take a look at this apple orchard. Can you see anything wrong with these trees? Anything in their size or shape? Now look again, like a farmer would. If you have a limited amount of land to grow apples, these trees are much too wide. They simply take up too much space compared with the amount of fruit they produce. But if they grew shorter branches, you could plant more trees on the same plot of land. Today, you can. Chemically engineered growth hormones actually make apple trees grow shorter branches. With plants, animals, and any organism, engineered solutions are nothing more than possibilities until they work here, in nature's testing chamber. As science opens new doors of understanding into genetic chemistries of life, how carefully we discover is just as important as what we discover. In 1975, scientists from around the world met to discuss guidelines for laboratory genetic testing. At that meeting, a group of DNA experts from the National Institutes of Health in Bethesda, Maryland, were asked to oversee genetic engineering experiments. They have ever since. Now, yesterday's controlled experiments are ready for real-world testing. But their first meeting with Mother Nature must be safe for everyone and everything involved. Today, our U.S. Department